Thank you for joining us for Nematode University, presented by Syngenta. I'm Julianne Johnston with Morgan Myers, a GNS agency, and presenting today we have Dylan Mangel, plant pathologist with the University of Nebraska, who is also a leader of the SCN Coalition. He will provide an overview of the SCN resistance problem and recommendations for active SCN management. And with Syngenta, we have Katie Jaeger, Seedcare Product Lead, and Dale Ireland, Seedcare Technical Product Lead. They will share some exciting news they have about a new product launch in the pipeline. We have allowed for ample time to answer questions after the presentations, so throughout the webinar, feel free to enter your question into the Q&A button near the bottom of the screen. But before we get started, we have a question for you. The question is, have you ever sampled for SCN? Yes or no? You can answer yes if you've ever pulled a sample for SCN any time of the year. That means sampling in the spring before planting or after harvest or any time in between. The SCN Coalition says soil sampling is the foundation for an active SCN management plan. So I think we'll give a couple more seconds for you to answer the question. And as soon as you feel like we have the answer, it will show up on our screen. And the poll shows that 60% of you have sampled for SCN sometime in the past. So Dylan, what do you think about the results of our first poll?
I think that's excellent. I mean, it's good to see so many have, but I expect a good number from a group coming to a talk on soybean cyst nematode, for example. But I'm interested to see a lot haven't, and hopefully we can convince you today with some of this information.
That's
Okay,
great. Yep, go ahead, Dylan. You can get started.
thank you so much. So thank you so much for everyone for coming out today and hoping to learn a little bit more about soybean cyst nematode as well as new management options. The reason we're here today is because of this number on the screen. Soybean cyst nematode is the #1 biological pest of soybean. It impacts soybean somewhere in the range of $1.5 billion a year in the US. So it has a humongous impact. But there's some problems with how we identify this and how we manage it. Slide, please. So I'll just start off by sharing a little bit of the biology. And I've outlined this in pictures. I'll start here on the top left, and the pictures are fascinating because most nematodes are too small to see. And in fact, soybean cyst nematode is too small to see basically for almost its entire life cycle. But in an area it's different from other nematodes, there's a small part of this life cycle where you can actually see this with the naked eye. Now, in that top left image, that's a picture of a soybean cyst nematode egg or several eggs. And if you look closely at those eggs, you can see it basically has its worm shape inside of there, folded over itself three or four times. From that egg, it's going to hatch. In that top middle panel picture, you can see a wormiform nematode that's going to stay in the soil. One key component of that nematode, if you look on the right side of the picture, you can see its mouth parts, and it has a stylet that's going to enable it to penetrate roots. And that's what makes this and other plant parasitic nematodes similar, is their ability to actually live and enter those roots. If you look at the next picture, you can see soybean root, and you can see nematodes inside of there. So basically, these eggs are going to hatch, and these wormiform nematodes are going to move towards that root, and they're going to infect that root. If we move to this bottom set of panels, you can see where the nematodes have entered the root, and they've started to modify that root. Now, these nematodes, they can go and they can basically try to feed on a lot of different types of roots. But it's only when they come into contact, or it's primarily when they come into contact with soybean, that something special happens. They've developed the ability to trick the soybean root to modify itself in a way where the root is going to start, that soybean plant is going to start feeding resources to that a nematode. They make basically what's a feeding site, and the soybean plant is just going to start channeling resources to those nematodes instead of to the seed or instead of to reinvesting that in plant growth. So these are small worms basically that are parasitizing, sapping resources away from those plants. They'll do that for a period of time, and then the males will sort of regain their worm form status and leave the root. They will go, and at this point in time, the females start to protrude from those roots a little bit. The males will fertilize the females. And you may ask, what do these nematodes do with all of those resources that they're sapping from the soybean plant? Well, they invest them in making eggs. So in that bottom right picture, you can see the females have started to grow as they basically expand and become full of eggs, averaging about 250 eggs per female. Now that's a lot to go from a single egg to 250 eggs in one nematode, but it doesn't just happen once a year. Their life cycle can complete in about 30 days. So depending on which region of the country you're in, they can easily have 3 or 4 or more cycles of this in a single year. So their numbers grow extremely rapidly. Slide, please. Here's just a starter on some of the signs and symptoms of soybean cyst nematode. Now, stunting is what we really think of with soybean cyst nematode. Um, you can see that in that right picture, but I'll get into that a little bit more. If you do see an area like that and you pull up and you carefully pull up roots, you might get lucky enough to see those nematodes on those roots. Now, this is easier done in sandy soils. Those nematodes want to stay in the soil, trust me. So when you pull them, they'll fall off if you're not careful. But if you excise those roots and wash them gently, you might see what's marked here with red arrows on the left. And next to a nodule marked with this blue arrow, you can see and kind of get an idea of the size. Now, my eyes are scarcely good enough to see these, uh, but even with a magnifying glass, sometimes you can actually see them. And with any form of magnification, they become quite visible. So each one of those red arrows is pointing at a female cyst. Now, the cyst is what happens when the female expands and becomes full of eggs, and then she dies and hardens and basically becomes a home for all of those eggs until they encounter more soybean roots and hatch. But they sort of look like small lemons colored structures. And if you're lucky, you can see them, but you can't rely on this alone because, like I said, they want to stay in the soil. Those are barely hanging onto those roots. So you've got to be very careful when you pull them out if you hope to see them. Slide, please. I talked about that stunting, but here's the insidious thing about soybean cyst nematode. Only in extreme situations are you really going to see stunting or any foliar or above ground symptom with soybean cyst nematode. If you look on the right, these are fields that actually showed visible symptoms, and those and under those varieties, they saw something like 15 to 50% yield reductions. So huge amounts of yield reduction. But on the left, it's very common. I'd say it's much more common to be in this 0 to 30% yield reduction range where you just have good looking beans a lot of the time, and you don't realize that you're losing that yield to the parasites in the roots. Slide, please. So that's the biggest issue, just figuring out this is there. And once you've figured it out, um, you can experience large ranges of yield loss just based on how many are there and what's going on. But you can't really picture that and quantify that without doing soil testing. So here's a figure representing the management recommendations of the SCN coalition. And the first is that soil testing. Right now is soybean cyst nematode action month, as I like to call it. It's one of the best times of year to sample for soybean cyst nematode. The reason is because in those soybean fields all summer, the nematode populations have been growing. So right now, if you've just harvested or you're about to harvest a soybean field, those nematodes are at their peak level in that field. If you go now, get a snapshot through a soil sample of what's in there, you're going to have a good idea of sort of your max level. Now, unlike a lot of corn nematodes that will move in the soil profile throughout the year, soybean cyst nematode is always going to stay in that top 6 to 8 inches of soil. So it's remarkably easy to sample for this. Any time of year you can go out there. populations are at max levels now. They're going to stay pretty consistent through spring. So anytime now through spring, you can get out there and get a good sample. But if you're just curious if you have this or not, any time of the year is a great time to start. Aside from soil testing, and once you know you've had it, the action threshold for soybean cyst nematode is 1 nematode. That's because they can reproduce so fast and grow to large numbers extremely fast. So if you have any of those, if you have any, you need to think about management. And the foundation for all soybean cyst nematode management and a lot of disease management in general is selecting a resistant soybean variety. So seed catalogs will list resistance types, and we recommend using those and rotating which ones you use. I'll talk a little bit more about that later. The next thing is rotating to a non-host crop. I think everyone in the Midwest should be thankful corn is a non-host crop. Corn does a huge amount of management for us in keeping those numbers low, breaking up that cycle. And although it's never going to get rid of soybean cyst nematodes in those fields, just rotating for 1 year to corn can help knock those numbers back by quite a bit. Rotating out of soybean longer has significantly diminishing returns, but a single year, potentially 2 years out, can really help bring those numbers down. And then last, we recommend considering a nematode protectant seed treatment to pair with those other management tools as well. Slide, please. So several states, many states across the Midwest, have free soybean cyst nematode testing programs, often funded through soybean checkoffs and in partnership with local labs or university labs. I'll share some results from Nebraska here in a minute, but what I just want to say is once you get a sample sent in and you get a test back, the test results are going to be reported to you in terms of the number of nematodes per 100 cc's of soil. So basically a half cup of soil and how many nematodes you have in there. But how do you interpret what that means to you? And it's quite a complex answer. I can't tell a grower exactly what 1,500 nematodes per 100 cc means to them because it's different for every soil type. It's different with pH. It's different with nematode populations. So what we've done here at the SCN Coalition and just released last year is a tool called the SCN Profit Checker. And this is a tool built on 25,000 plus plots of data that we've collected over many years that helps estimate what sort of yield loss you would be losing if you had a certain amount of eggs in a certain type of field. So in this case, there's 1,500 eggs per 100 cc of soil. There's another line here called the female index, which is auto-populated based on state research averages. But basically, this is showing how well those nematode populations can reproduce on specific resistance types. And I'll talk a little bit more about that later too. Sand percentage plays a huge role. As sand increases, nematode activity increases, and pH likewise. So if you know your field's sand and pH, you can get a good idea of yield loss from that. And this situation estimated a 5.7% yield loss to soybean cyst nematode. Slide, please. Now, SCN and damage from SCN is growing. It's really hard to picture this when you have a pathogen that's not showing above ground symptoms in most situations. But there's two factors that are increasing the amount of damage caused by this. The first is just expansion of the nematode to new fields. Now, this is a figure we just put out last year, led by Greg Tilka at Iowa, which shows infected counties, and those are in red, and blue are counties that are newly infected. So it's just been reported there for the first time. Now, a great point on this slide, though, is those red counties represent most of the soybean producing acreage. We're not going to have it in western Nebraska, for example, where we don't grow soybean. So a lot of the acreage has really, it's spread to those counties, but just because it's in those counties doesn't mean every field has it. Slide, please. If you look at Nebraska, for example, this is the last 10 or so years of our testing program. These are the positivity rates across different counties that we see, ranging from 0 to 50 or 60%. So basically half the samples in most of these counties, or far more than half the samples, are not positive for soybean cyst nematode. it's still spreading. So in the US, if we're losing that 1.5 billion to soybean cyst nematode, there's still a lot of upside as that's going to continue to spread from field to field until ultimately almost every field will be infected by soybean cyst nematode in time. That's the first reason this is getting worse. Slide, please. The next reason that damage from soybean cyst nematode is growing is because the resistant sources that we are using to protect from soybean cyst nematode are breaking down. We have very limited tools available when it comes to these resistant sources, and they're not as effective as they used to be. Slide, please. The two primary resistance genes available in varieties on the market right now are called PI88788 or peaking. Now, PI88788 was added to varieties widely in the late 1990s and continued to be added until, for a time, almost every soybean variety in many maturity groups contained PI88788. Even to this day, the huge majority of varieties contain PI88788. And you can see that if you go to the catalogs, it will tell you which resistance gene they have. In more recent years, PI88788 has experienced reduced effectiveness. Basically, it worked really well for a long time, but what happened was we overused this one management tool. We kept planting it into fields, and those nematode populations basically evolved and learned how they could reproduce on these varieties despite it being resistant. So we'd seen where these might have been 90 or 100% effective to become 80, 70, 60, 50% effective. Now we've seen fields where they might be 10% as effective as they once were. So while this resistance source is still effective in many fields, it's rapidly degrading to become less effective, but still useful. On the other hand, peaking is a resistance source that started being added in the early 2010s into varieties as 88788 was diminishing. And it's taken time. The breeding process is very slow, and it's taken time to get this peaking resistance gene into really good backgrounds. But breeders have been at that for over a decade now, and now we're getting peaking varieties that are performing extremely well. So if you haven't tried peaking, or if you haven't tried peaking in several years, try planting a peaking variety in those fields and seeing how that works. But just like 88788, do not rely just on peaking. Peaking works better than 88788 ever did. And how that's going to translate to resistance durability is the nematodes are going to overcome it even faster than they did on 88788. But what we now have are two effective tools, and if we rotate them effectively, they're going to last as long as possible. So maintain these and protect these by rotating between different varieties. Slide, please. So I just want to give a demonstration using the profit checker on how that resistance breakdown is impacting a specific field. So in Nebraska, the median number of eggs per 100 cc's of soybean cyst nematode that we're testing in our programs is 4,000 eggs. I'd say that's a moderate number. If you've got below that, keep managing effectively. If you've got above that or much more than that, we really need to talk about how we're managing and try something different. In that first example, the state average for my state for the female index on PA88788 is 35%. So those nematodes can reproduce on that resistance source about 35% as well as they can reproduce on a susceptible variety. So it's still working quite well in this case, but not nearly 100% effective. We'll use 20% for sand and 6.5 for soil pH, and we get a yield loss of 6.1 in this case. That's a pretty good, average for Nebraska. That's a big reason why this problem often goes unchecked is because if 50% of fields are losing 6.1%, you might not notice it. You might not miss it if that's just what you're used to. But what happens is that resistance gene is going to continue to break down. Yield loss is going to increase. So we'll use the profit checker again on the right side, 4,000 eggs. Now I've changed the female index to 90%. That's not uncommon. We've seen that, and we're seeing that more and more. We've had female indices into the 90s, well into the 90s. Same sand and soil pH. And if you could click the slide, we can see what yield loss we're losing from that. So in the same field, after continued use of that same resistance source, the nematodes are evolving, and now you're up to 15.5% yield loss. This isn't going to happen fast. We're slowly creeping up from that 6.1 to that 15 and more. And one thing I didn't factor into this is as that female index goes up for 88788, your egg population isn't going to stay at 4,000. It's not uncommon to see 20,000 eggs per 100 cc. I've seen 100,000 eggs per 100 cc. So it does get a lot worse than that 15.5%. But as female index increases, it's a huge impact. So despite this nematode being widespread in our country, there's still a lot of potential for this to get a lot worse than it is right now. Slide. So I'll just finish on this slide. I wanted to get my point across that we need to manage for soybean cyst nematode. It's hard to tell the difference between that 6% loss and that 15% loss. And it's hard to know if your varieties are working and your rotation is working and your seed treatments are working without testing. So take advantage of testing programs in your states. Call your soybean checkoffs and call your state extension services and find those programs in your states. And the last point I want to make is we need to use every tool that's available. If we focus and just use resistant varieties alone, they're not going to last. If we use the same tools over and over, they're not going to last. So consider a holistic approach, use everything we have. That's the point of the SCN coalition here is I think everyone agrees that the problem isn't what management you're using. The problem is people aren't using management. So it's an awareness tool. With that, I'd just like to hand off this talk to Katie Jaeger with Syngenta, who's going to talk a little bit more about a new tool that they have available.
All right. Well, thank you, Dylan. I hope you guys got as much out of that presentation as I did. It was very insightful. And before I get into the content that I have to share with you this afternoon, again, Katie Jaeger, part of the product marketing team with Syngenta, really grateful to be here this afternoon. And Dylan, happy belated National Nematode Day from Monday. And just wanted to thank you guys as a coalition for your commitment and dedication in this space. Not only for being at the forefront of awareness for SCN, but also being at the core of the promotion of active management strategies and tools to help prevent yield loss. Because when you think about the purpose of all of us, whether you're a panelist like myself or an audience member tuning in this afternoon, the fact that we know is that as nematode reproduction increases, yield decreases. And collectively, that's our goal is to find management strategies to increase productivity on the acre that results in higher profitability, which is our core objective for you guys. And with the changing dynamics that he eloquently just voiced over, with having a single source for resistance for the SCN populations becoming resistant to the resistance, that then drives the need for new technology to further address some of these evolving needs. So I'm excited to share some new innovation from Syngenta. We have a new seed treatment coming for the 2026 planting season. The brand name is Victrato. The active ingredient is TYMIRIUM technology. And to voice over high level what this product is and what we're going after, if you look at it on the screen in front of you, obviously, if you look at the value proposition, nematodes are core to the value proposition of this product. We are seeing high levels of efficacy against a broad spectrum of nematode species, but in addition to that, you will see some key diseases in soybeans also on the list. If you look at the active ingredient, you probably don't recognize it because it's new. This is new innovation that we're bringing to the market. It's cyclobutyflurium, which of course is not easy to pronounce, nor probably is it easy to remember. So we've taken a strategy of branding the active, and we'll refer to it as TYMIRIUM prior to registration again with the brand name being Victrato. after approval. You might recognize the mode of action. This is an SDHI. If you think about brands that you might recognize like Saltra or Levo, they also sit in this class of chemistry. So these SDHIs go after energy inhibition within the cell, and Victrato will sit in this class as well. So on the point of new technology, if you're think about bringing a new product to market, I'll let a poll here show in just a second, but how long do you guys think on average, if you guys can weigh in, how long do you think it takes to bring a new product to market? We'll give you some choices and let you weigh in here. The choices are 1 to 3 years, 4 to 9, or 10 or more. We probably should have put forever on this list because that's maybe how some of you feel. But on average, wanted to get your opinions and let you weigh in. Okay, so we'll show the results. And my guess is that you're probably exactly right. Yeah, okay. So some of you are more optimistic maybe than myself today. Yes. 4 to 9, some of you weighed in on that, but the majority of you, yes, it is 10 or more years. On average, it takes about 12 years to bring a product to market. And, on the next slide, we can go through the journey that has led us to the point we are today. So a lot has been involved in the process of bringing this molecule to market. It actually began in '08 when the molecule was first discovered. The first field tests were done in 2013, so about 12 years ago. So an extensive amount of field testing done here in the US with the submission in 2021, awaiting registration here today in preparation for 26. In total, over 300 trials have been done. So we have extensive knowledge and experience with the performance of this product in the US market. And to put this into dollars, on average, it takes about $300 million to bring a single product to market. So hopefully you can see our dedication to innovation and bringing new technology to the market. So with that being said, let's dive into specifically what we're going after. First on the list, you'll see sudden death syndrome. So again, if you think about the products today, the foundation of bringing these SDHIs to market was actually all around sudden death syndrome protection. And the unparalleled performance that we're getting with Victrato is something that we're very proud to bring to you as a customer, knowing the issue that this is for a lot of you. Moving to nematodes, we know the correlation, right, between nematode populations and sudden death syndrome infestation, with the nematodes being the vector for the fusarium to enter. Victrato is broad spectrum again. Soybean cyst nematode gets a lot of the light, as it should, because it is the most prevalent. However, in some of our southern geographies over into the eastern seaboard, up into the mid-Atlantic, we see some other species, including root knot, and is equally highly efficacious as on the other species as soybean cyst. Next, you'll see that this product will be the first with a federal label for suppression of red crown rot. So red crown rot, briefly, it was discovered in the 70s. However, it was identified in the Midwest in 2018 in Illinois. The Crop Protection Network has done an excellent job of tracking the incidence. And the reason why this is important is first of all, there are limited solutions for this disease at the moment. And secondly, when you think about the infestation of the disease, again, with sudden death as the frame of reference, that can cause anywhere from 20 to 40% yield loss. And it's actually more destructive when you see an infestation of red crown rot being upwards of 50%. So very excited to have that included on the label. And then lastly, early season suppression of the foliar diseases. Frog eye, leaf spot, and target spot will be the new additions versus what's on the market today. So no matter the category, the performance is really unmatched. And we're able to experience a new level of protection that we haven't seen from a product on the market. And it really boils down to the molecule itself. The level of potency that we're able to achieve then correlates to the level of performance that you guys will witness in the field, and what Dale will allude to during his presentation. So I've got a brief video to really illustrate and bring this to life. not only showing the destruction of some of these targets on root development, which is really the impact that the targets have on yield. And this video not only illustrates the impact but the benefit of the seed treatment. So I'll share this with you, and then I'll turn it over to my coworker to really give us some reasons to believe and data behind the performance. So thank you again and enjoy the video.
Every soybean grower knows the battle they face at planting. The journey begins as soon as the seed is planted. Almost immediately, the battle lines are drawn as nematodes and pathogens begin their attack. As seedlings take root, nematodes work to breach the vulnerable root structure. When nematodes damage the roots, they open the door to many other pathogens, which causes diseases like sudden death syndrome.
Every year, U.S. soybean growers will lose over $1 billion in yield to soybean cyst nematode alone, and other species will steal millions more. But every challenge becomes a learning opportunity, a chance to understand the unseen enemy's attack strategies. Soybean cyst nematodes move to the vascular tissue to establish a permanent feeding site that damages the root. Root knot nematodes penetrate roots and release secretions that form feeding sites, which then result in galls, restricting water and nutrient uptake. Lesion nematodes move in and out of the roots as they feed, creating necrotic lesions that damage roots and open pathways for pathogens. Reniform nematodes penetrate the root cortex and establish a permanent feeding site, limiting root function. Lance nematodes feed both internally and externally on the roots, destroying root cells, limiting water and nutrient uptake.
Meeting this onslaught of nematodes and pathogens head-on helps establish the crop. Syngenta has developed a seed treatment featuring TYMIRIUM technology, coming soon. Let's learn more about its technical attributes. To explain the science behind this novel active ingredient, we turn to our expert.
During our research, we capitalized on our deep understanding of carboxamide SDHI molecules and their functionality to design a molecule that adapts a single shape that reaches high potency against very specific targets.
As we take a closer look with its molecular design, it can only bind to target sites on nematodes and soil-borne pathogens like Fusarium, leaving other organisms, like beneficial bacteria and earthworms, untouched. It will provide protection against labeled pests without damaging the soybean seedling. Observe. A healthier root system leads to greater resilience later in the season. TYMIRIUM will deliver early season suppression of foliar diseases like frog eye leaf spot, target spot, and Septoria brown spot.
It will protect against nematodes and diseases, helping to preserve green leaf area, enabling the plant to realize the benefit of seasonal inputs, like foliar fungicides.
TYMIRIUM technology, coming soon.
Good afternoon. This is Dale Ireland. I never get tired of seeing that video, I have to say. Pretty cool illustration. I want to thank Katie and Dylan for their portions of this talk, and appreciate your time today. I lead field development efforts for Syngenta Seedcare for corn and soy seed treatments, new molecules, new combinations of molecules This is the 11th season that I've had replicated trials in the field on soybeans. So it's about time for this thing to get registered, and we're very excited to be sharing some of these data with you. Next slide, please. Reinforcing what was just mentioned in the video, TYMIRIUM is a very safe molecule across the rate range. There's a single rate that we're offering in the US. That's the 50-gram rate, or that 0.714 fluid ounce per 140K unit. And at that rate, and even higher than that rate, it's very, very safe. So I just really want to reinforce that compared to the leading competitor. Next slide, please. One thing that both Dylan and Katie have mentioned is just the protection that TYMIRIUM offers. And this is something that we've really been trying to build out. We have a lot, we have a decade worth of yield data. But recently we developed a root scanner app for iPhones. And what we showed last year, nearly 2 months into the growing season, and keep in mind, this is only at 1,400 SCN eggs per 100 cc's of soil. So about 1/3 the amount of SCN that Dylan was talking about at 4,000. So at 1,400 eggs, what we saw was that TYMIRIUM protected 33% more root surface area or root fresh weight than the check treatment. So Cruiser-Max Apex, bar on the left, and then you have TYMIRIUM, the bar on the right, on top of that same base treatment. And what we did was we protected 33%, or 1/3 more root area if you looked at an average of 21 root masses. So I don't know a grower out there or a retailer out there that isn't interested in protecting root surface area or root mass. And we don't know what 2026 will bring. If you use Victrato, what these data would say is that statistically different level of root protection you receive from Victrato, or excuse me, TYMIRIUM. So that was number 4 of those 4 different ways you can try to manage SCN that Dylan talked about. So let's see how that translates into yield. Next slide, please. So this is a data table of many, many data points. If you look at the first row there, you see TYMIRIUM at that go-to-market rate. So it's TYMIRIUM plus the base treatment against just the base treatment alone. We call that the check. 124 locations. Each location would have 4 to 6 reps. You see that in that center column, 124. And then 10 years of data, 83% of the time, TYMIRIUM will beat, or Victrato will beat, the check treatment by between 3.5 and 4 bushels per acre. And then the second and third rows, that's FLPM, that's the abbreviation for the active ingredient and the leading competitor. Fluopyram is the active ingredient. The leading competitor is Alivo. I believe most of you are familiar with that product. The first or the second line there at the 0.075, that's their nematode only rate. The third line across is their SDS rate. And you can see in both cases, TYMIRIUM handily beats it almost 3/4 of the time, between 2 and 3 bushels. And again, all of these are under moderate to heavier SCN pressure. And then that bottom row is abamectin, ABA. That's Avicta for some of you in the South. We've marketed that seed treatment for many years. It's not as popular in the Midwest. So needless to say, for SCN, that's what all these data are for, that we are protecting a lot of yield, anywhere from 2 to 4 bushels under these conditions over the last decade. Next slide, please. Shifting gears to another nematode that Katie had just talked about, root knot nematode. In this table, a bit less data. Again, it's set up the exact same way with the exact same treatments. A lot fewer locations, just simply because there's not as much root knot when you look at all the acres across the US, but there's still a significant number that do have root knot. And you can see that between 65 and the mid 80% of the time that TYMIRIUM beats the check treatment, whether it's the fungicide check alone at 86% wins and about 5.5 bushels of yield difference between the base treatment compared to TYMIRIUM plus that same base treatment, or the second row there, over 80% of the time beating the leading competitor by between 3.5 and 4 bushels. And then that third line, that's at that higher SDS rate of the leading competitor. That's about 65% of the time between 1.5 and 2 bushels of yield advantage over the competitor. So needless to say, we're very pleased with the performance, whether you're looking at SCN or root knot nematodes. And I assure you the other species would be much the same, if not better. Next slide, please. So this is a data summary of 9 locations, 6 reps at each location. This is back in 2021. And the reason I'm showing you this is just that it has several locations: Iowa, South Dakota, Nebraska, Minnesota, several in Illinois and in Michigan. And we're comparing the base treatment on the far left side to, and all the other 4 treatments all have that same base treatment underneath it. You have Saltro at the go-to-market rate. You have TYMIRIUM in the center, the blue bar, and then you have the 2 rates of the leading competitor. And what we see are all of the treatments are better than the check treatment. You can see that by the statistics. The check has a C as in Charlie next to the numeric figure. Then you look at Saltro and the 2 rates of the leading competitor. They all have the letter Bravo or B beside them. That means that those are not statistically different across these 9 lokes back in 2021 that have moderate to heavy SCN pressure. And that just means that if you looked at these treatments again in 2026, you could have a 90% confidence level that you're going to see a similar performance. And really, the reason that I'm showing you this is simply that TYMIRIUM sets a new level of protection against nematodes in the field of anything that's offered, whether it's one of our internal products or whether it's either rate of the leading competitor. Predictably, TYMIRIUM is going to be the leader with a 90% confidence level. Next slide, please. Let's shift to SDS. As Katie had talked about, that's also on our label and a very impressive showing here. This is TYMIRIUM at the go-to-market rate versus the leading competitor at their SDS rate. Over several years, we beat the leading competitor 91% of the time by between 4.5 and 5 bushels on average. So again, each one of these are replicated locations. We're across several years here. I think if any one of us had a 91% win rate in anything or any pursuit we were doing, we would bet that that was a very good decision to make. So again, I've shown you nematode data. This is SDS data against the leading competitor. Let's see how this product does against Saltro. And that's what this slide is showing. So this is TYMIRIUM at the go-to-market rate, Saltro at its go-to-market rate. 87% of the time, TYMIRIUM is going to beat our leading product. The leading commercial product today, based on many years of data, is Saltro. And TYMIRIUM sets a whole new yield level at 87%. That's a statistical difference. The next time you see these two, you can count on TYMIRIUM beating Saltro. And you notice that the yield difference is about half what it was compared to Ilevo, however, at between 2.5 and 3 bushels of difference rather than 5 to 6 bushels of difference. Let's shift gears here to the next pest that is on that list. Next slide, please. Let's look at red crown rot. Many of you are familiar with red crown rot. Hopefully you've seen this on someone else's field and not your own, but if either way, just know that TYMIRIUM is going to be your best tool against red crown rot as far as a seed treatment. You notice on the far right-hand side of this slide that that is just a fungicide insecticide based treatment. That's Cruiser Max Apex, our leading fungicide insecticide, not registered for red crown rot. On the left-hand side, those nice healthy looking beans. The only difference between those two strips is the fact that the strip on the left-hand side has TYMIRIUM on it, and it protects a lot of yield. Now, why would you expect the strip on the left-hand side to look so much different than the one on the right under heavy red crown rot pressure? The next slide, go ahead and advance that, will show you that basically on that right-hand side of that previous slide, they basically have no roots left. And that's what red crown rot does. And that's why it's so devastating, is it just destroys that root system, even worse than SDS does. And you just basically have no roots left. And you can see how much TYMIRIUM protects that root system. And these are just average plants from that very plot. And you can see how much more root system that those have. And that's why those soybeans are so intact. It's holding all those pods. It retains the flowers, and it protects additional plant yield. As Katie mentioned, it can approach 40 to 60% yield loss with red crown rot without even, that's not even a worst case scenario. So red crown rot's the real deal. Next slide, please. Now we'll look at some actual yield data. This is 4 locations from last year summarized in bar charts. You can see each one of the locations there as different symbols, different colored symbols on each bar. On the far left-hand side is our fungicide insecticide base treatment. That's under the rest of the 4 treatments also. We have Saltro at 2 different rates, 50 grams and 100 grams. Then the second bar from the right-hand side is the TYMIRIUM treatment. And then you'll see the leading competitor at their red crown rot 2EE rate. Now note that Saltro has a 2EE at either one of these rates, the 50 or 100 gram rate, but that either rate of Saltro or that very high rate of the leading competitor, they're all in that Bravo statistical class, or B as in Bravo, just meaning that you would not expect any one of those 3 treatments to act differently the next time you see them. You'll notice they're all better than the base treatment, which is the Charlie group or the C statistical group. And then again, as I've reinforced with nematodes as well as SDS, TYMIRIUM sets a whole new yield level against red crown rot also. This is truly a Swiss Army knife. It really is a very good tool for many different pests. But TYMIRIUM sets the highest yield level that we have seen across years in multiple lokes against red crown rot. Next slide, please. So in summary, what I've shown you in the last few minutes is that trial performance has shown better results from TYMIRIUM than any of our internal or external checks. And we've taken only the best products that are out there, the products that perform the best for us under nematodes and under SDS. And under SDS, it offered between 4.5 and 5 bushels with a 91% win rate against the leading competitor across multiple years, many locations, and had a very nice performance. And then under red crown rot pressure, nearly an 8 bushel difference with 100% win rate against that same leading competitor at their 2EE rate last year. So again, obviously, we're going to have additional data in 2025, and we look forward to sharing that with you when we have it. I think at this point, I'll turn things back over to Julianne, and thank you again for your time.
Thank you, Dale. We do have some questions, but there's still time. If you'd like to enter your question, there's a Q&A button at the bottom of your screen, and I will welcome our panelists to turn their mics on. Our first question that came in today is for Dylan. And if I'm not planning on growing soybeans in a specific field next season, do I still need to test for SCN this season?
Yeah, great question. When we're, a lot of fields are on a 2-year rotation. In Nebraska, we got some on a 3-year rotation. You can test at any point in that rotation. and you'll have an idea of what's there. Nematode populations will fall. If there's a non-host in there, say corn, for example, you could expect those populations to fall from their peak 50% in 1 year of corn. So if you harvested after that corn season, because you're going to put beans in there the following year, you'd know if you have it, but you'd have to account for that point in your rotation to sort of anticipate how many nematodes were actually going to be there or were there after your last season. So even if you're finishing with beans and going back into corn or something else, it's just a great time to pull that number because you know your peak level. That's sort of a checkpoint. Now, if you do want to sample after corn, for example, just before you're putting beans in every time, and you want to consider how those levels have changed over years, make sure you're comparing numbers that were sampled at the same point in that rotation. So as long as you're comparing apples to apples, you can make and get a good idea of if your management is effective.
And here's a follow-up for you, Dylan. Once I learn that I have SCN in a field, how do I eradicate it?
You really can't. It's extremely difficult. Now the nematodes are very smart. They have evolved to respond to when soybean is present in that field. So if you're not planting a soybean, they're just going to wait in that cyst form in those eggs, and they can wait for up to a decade pretty easily. So they can last a long time. The other big problem is they have some alternate hosts, not corn, for example, but some weed species will host them so they can maintain themselves even if you say didn't plant beans for 10 years. You'd still have them there. So you can't eradicate them. You just got to work with what's there and use management tools and rotate those tools to make them last as long as possible.
And Katie, our next question is for you. Noting that frog eye is on your label for early season, is the frog eye that I'm seeing in the Midwest considered as an early season infection, or is it coming in later during the growing season?
Yeah, thank you for the question. This is an excellent question. No, and maybe I'll start with this that TYMIRIUM will not replace a foliar application for any of those foliar diseases, whether it be septoria, frog eye, or target spot. What we're seeing is that with the TYMIRIUM treated seed, it is putting the plant in a better position to receive the foliar application. So we are seeing lower disease severity ratings early, and then combined with the foliar application is really the 1-2 punch to get you a higher yield trajectory than either one of them alone. With the potency of the product, it is still a seed treatment, right? So though we're able to get levels that we haven't seen before, our concentration is and will remain that first 30 days. Dale, I'll let you weigh in if there's anything I missed, but that's how I would address the question.
Yeah, I would just reinforce what you said, Katie, but also just the fact that when you do apply your foliar fungicide at R2 or R3, your plants will be significantly cleaner, allowing a higher yield trajectory from using that system of TYMIRIUM as well as the foliar fungicide.
And Dale, this next question is for you. What are the key considerations for integrating this new technology into an overall SCN management plan?
Yeah, so let's reinforce what Dylan led off with. I think that's really important. That was sampling, right? You've got to know whether you've got the critter or not. And most people do. If you've been growing soybeans, or if your farm's had soybeans, or your customers has had soybeans for decades, you generally do have SCN. Once you have it, as Dylan also said, you're never going to get rid of it. And as our great friend Greg Tilka from Iowa State always says, keeping low numbers low is important. So sampling, then making sure you use resistance and rotating that resistance and rotating to non-host crops. But what you can do is stay up with the very best solutions for seed treatments in addition, because we want to use all the tools in our toolbox, right? All 4 of these tools that Dylan introduced us to. And TYMIRIUM is going to be in that final bucket. And that is making sure that you use Victrato when it's registered will be the name of the product. And you can't buy a better nematicide across the labeled products that are on the market today And this sets a whole new level. So as far as using the sharpest, best tool, you're using the best rotation practices with non-host crops for a year or two between soybean crops. You're using resistance in your soybean varieties, but then you're also using the best soybean nematode and disease products, such as TYMIRIUM, as a seed treatment. That's really all you can do.
And then we have another question for Dylan. How often should we advise growers to rotate Peking and PI 88788 resistance?
That's a great question. It's excellent to have more than one choice on the market, right? Uh, and we're recommending rotation all the time. So I would never plant PA 88788 after itself, and I would not plant Peking after itself. The problem is, though, I think a lot of growers are going to have an excellent experience with the new Peking varieties. They're going to have fields that have gotten used to 88788. They'll put a Peking and they'll discover some yield that maybe they didn't know was missing. That could create a lot of short-term incentive to follow that up with another Peking after next time you rotate into that field with soybeans. Resist that urge, though. Peking's going to degrade faster than PI 88788 ever did. So, but if you rotate them, we can maintain both of their effectiveness as high as possible.
I have another question for you, Dylan.
Mhm.
You mentioned that SCN and SDS have a working relationship. Are there any other crop diseases that SCN amplifies?
Well, when you start off again with an unhealthy plant, when you have a diminished root system or a root system that's not as effective, and you have those nematodes have made a sink for the plant to be sending resources down to those roots. The plant just doesn't have the resources to fight as much of anything or as well on anything. So it's going to have some impact across the board. We see it most with other root rot pathogens, right? Because you have opportunities for those to get in there. And sudden death has been the most obvious of those. But really, when we're sick, if any one of us had a parasite in our stomach, we wouldn't be set up very well to fight off anything like a cold or anything like that.
Yeah, do Dale or Katie have anything to add to that?
Yeah, I think the only thing I would add to what Dylan said is that when you think about livestock, I mean, if you've got a steer that's severely parasitized, it's not going to perform. The weight gain's not going to be as good. I mean, it's pretty common sense, but you need to think about you're destroying root surface area, but then also you're stressing that plant in addition to that, and it's just not going to perform. It's going to abort more flowers, drop more pods, and that's where that yield difference comes from.
Which state in the Midwest has the overall highest percentage numbers of SCN from available soil sampling data? Does anybody have the answer to that question?
I'd say a lot of the states run their programs independently. So we've got generalized data on where they are, but it's hard to say who's got the highest. Now, what we do know is how this moved through this country. Like in Nebraska, we found it for the first time in 1986, and it's slowly spread west. And as it's spread west, it's infected more and more of those fields. Once you get over to the I states, Iowa, for example, their positivity rates are much higher than ours. It's just a time thing. So where it's been the longest and where there's the most soybeans is where we're finding it the highest. So somewhere in the I states, I would say is probably the highest, but we've still got this western front, Nebraska, Kansas, North Dakota, South Dakota, where we're kind of halfway across the state. And additionally, we've had a lot of added soybean acreage on this side as well. So there's fields that might not have it just because they're just getting soybeans for some of the first time as well.
Well, and Dale, I think we have time for one more quick question and answer. When do you recommend Victrato and when do we recommend Saltro?
That's a great question. Certainly in a red crown rot situation, Victrato, when you look at root knot nematode or a mixed culture of different nematodes, multiple different species, definitely Victrato. When you have SCN plus SDS problems, if you've got a history of a problem SDS field, I think that's another great Victrato. If you don't know whether you have a problem, or you don't sense, or your soil samples don't show that you have high levels of SCN, and you've never seen SDS in this field, Saltro may work fine in those cases. Certainly, there is basically any time that you have multiple pests that you want performance from against, Victrato is going to offer a superior. But I would also say that in no case of any of the data that I have, even when no pests are present, would Victrato be a problem or not offer great performance. So I guess I'll just leave it at that. And anytime that you have heavy pest pressure or multiple different pests, Victrato is the clear choice.
Well, I think we're at time, and we appreciate everybody for joining us today and wish you a very safe harvest season, and encourage you to visit the SCN Coalition's website for more information about active management, and make sure you visit the Syngenta website to learn more about the new product that is coming soon. Thank you, everybody.
Thank you.
Thank you.